Hey, my name is Christopher, and I will be presenting on the topic of inclusive fitness. The article that I've selected is Double-Edged Heat. Honeybee participation in the hot defensive bee ball reduces life expectancy with an increased likelihood of engaging in future defense. This study was conducted by, forgive me if I mispronounce these, Yamaguchi, Yugajin, Utagawa, Nishimura, Hattori, and Ono. And it was published into the Behavioral Ecology and Sociobiology Journal in 2018. The Asian honeybee and the giant hornet are natural enemies of one another. While the honeybee forages for resources to last the duration of the winter and allow for their queen, aka their mother, to reproduce, the giant hornet will prey on the honeybee hives housing these valuable commodities. The hard exoskeleton of the giant hornet makes it resistant to the stinging attacks of the bees, so the bees have evolved an alternative defense mechanism called the hot defensive bee ball to deter unwanted guests. This behavior involves a gathering of many bees around the much larger hornet, as seen on the right. While surrounding the hornet, the bees will rapidly vibrate their flight muscles. By doing this, they each generate a considerable amount of metabolically sourced heat, which then becomes concentrated in the center of the ball that is formed. The bees will do this until a temperature of 46 degrees is achieved over a period of 30 minutes until the invader succumbs to the heat. Most of the previous literature is pretty much focused on how well the bees are at eliminating unwanted invaders, such as the giant hornet, but there hasn't really been much consideration or investigation into what cost that this behavior has on the bees that participate. The authors state that there was one study that observed around a quarter of the bees die off after they had participated in a hot defensive bee ball, which I guess kind of got them curious on the subject. And then before we move forward anymore, I thought I should mention that the thermal limit of honeybees is roughly 50 degrees, whereas for giant hornets, it is 45 degrees, which is 1 degree less of the 46 degrees achieved by the hot defensive bee ball. So this study set out to determine if there's a potential cost that participation in this defensive behavior has on individual honeybees. This leads us into the next slide, the hypothesis. The authors weren't really straightforward with saying what the hypothesis of their study was, but I've kind of pieced it together as the following question. Is there a potential cost to the individual honeybees that participate in the hot defensive bee ball? So are they getting hurt or are they dying? To test this hypothesis, the experimenters set up 8 different observation hives at Tamagawa University in Tokyo, Japan, and populated them with the Japanese honeybee, a subspecies of the Asian honeybee. These hives were connected to a corridor that led to a field just outside of the laboratory so that bees could fly about and do bee stuff. The orifice of this corridor was vented with slits big enough for bees, but not hornets, as any hornets entering the observation hive would hinder the experiment. Hot defensive bee balls could be induced by presenting to the colony giant hornets that the experimenters gathered from the campus. All right, so here I've just added a slide so we can take a closer look at what's going on here. Here you can see the observation hive with the colony situated in between 2 glass panes that kind of act to prohibit the bees from escaping into this room. This bottom portion here is the corridor that allows the bees to come down and exit into either the field that's out there so they can freely forage and whatnot, or be presented the giant hornet through this little portion here, where the bee ball is ultimately observed. On the right hand side here, you can see one of the giant hornets attached to a small thin caliper like device that wraps around its body. I figured this image kind of isn't really that clear, so I've tried to illustrate it here. The experimenter can stand here, poke this hornet, I guess, into the hole, and then that induces the bee ball observed here. And then in this ball, you have bees climbing on top of bees, climbing on top of even more bees. And they're all just, again, rapidly vibrating their flight muscles, which generates a lot of like metabolic heat. And all of that heat gets concentrated on the hornet that is underneath of them all. I've included a quick little video here to show you how this is working. Apologies for the sound if that was loud. So yeah, here's the hornet attached to the caliper-like device. And then here's it being presented to a hive. And then the bees just go absolutely nuts. They just jump on them. And then that happens for around 30 or so minutes. And then after this, the experimenters can pretty much just lift the ball. This is still attached to the hornet dead body by this time. The bees are just kind of, they're still going hard at it, I guess. Yeah, this guy just kind of walks around with them. I'm not sure where he's bringing them to, but that's the video. Hope that helped better describe what's going on. All right, so this study consisted of 3 main experiments. The first experiment involved 2 steps that investigated survivalship following exposure to a bee ball, as well as various temperatures of incubation. So for part 1 of this particular experiment, worker honeybees were kind of grown or incubated at 32 degrees over a 5-day period. And during each day, the emerging bees were marked with different colors that the experimenters could use to see the age of the bees. 15 days after the final emergence of all those bees, the bee ball experiment was performed. So all the bees that were participating in this were between 15 days to 20 days of age. The experimenters presented a single giant hornet to the colony, which induced the bee ball. After 40 minutes, the experimenters collected the bees from the bee ball and color marked them depending on whether they were participating at the center or the periphery. An equal number of non-participating bees were then withdrawn from the hive and given marks indicating that they did not participate. And the experimenters then observed how many of each marked bee survived over the next couple of weeks. In part 2 of this experiment, bees were withdrawn prior to the presentation of the giant hornet that I just described. These bees were therefore bee ball naive, meaning that they did not participate in any bee ball defenses or really have any clue that one even happened. They then incubated 19 bees in each temperature, 32 degrees, which is pretty much room temperature for this experiment, 44 degrees, which is 2 degrees below the maximum temperature reached at the core of the hot defensive bee ball, and then 46 degrees, which is the actual temperature reached at the core. The bees remained in these temperatures for 30 minutes and were then returned to the hive for their survivalship to be observed over time. Moving on to the second and third experiments of this study, experiment 2 involved analyzing changes in heat shock protein 70 expression following exposure to different temperatures. Without going into too much detail here, the upregulation of heat shock protein 70 serves as a biomarker following cell death from exposure to high heat. Similar to previously described, experimenters removed 18 bees that had not participated in a bee ball. 6 bees were each incubated in 32 degrees, 44 degrees, and 46 degrees for 30 minutes, and then humanely executed. The brain, fat tissues, and flight muscles were dissected for RNA analysis, basically just determining how much heat shock protein was released following heat exposure in each of the relative areas where heat-induced cell death can occur. Experiment 3 was pretty fun. This one that mostly relates to the topic of inclusive fitness. For this one, they measured the bee behavior during a subsequent hornet attack. So to test this, a sample of bees were removed from the colony prior to any bee ball being induced. with some kept separate and marked as B-ball naive, and others incubated at 32 degrees, 44 degrees, and 46 degrees. A hornet was then presented to the colony inducing a bee ball, and after 30 minutes, the bees were collected from the ball and color marks depending on whether they were participating in the center or the periphery. And then this bunch of bee ball experienced bees, as well as the bee ball naive bees and incubated bees that were gathered beforehand, were all returned to the hive. The authors state that bees typically resume foraging behavior around 40 minutes-ish after an attack has died down. Therefore, they waited around an hour after the end of the previous bee ball before presenting a second hornet to the colony and inducing the subsequent bee ball. The bees in this ball were again collected and given marks depending on participation around the periphery or center. And then the participation rates of naive marked bees and bee ball participant bees within the second induced bee ball were measured. And then on to the results. For the first experiment that I described, it was found that participants of the hot defensive bee ball were significantly more likely to die earlier than those that did not participate. This can be seen in the graph on the left. There's a rapid die-off of bee ball participants within the first couple of days following their defensive behavior, whereas non-participants exhibited a slow kind of paced decline. And then on the right here, this figure displays the survivalship over time of the bees incubated at the various temperatures. Here you can see that bees incubated at the high heat of 46 degrees, which is the temperature achieved at the core of the bee balls. These bees died much, much sooner than those incubated at the not too much lower 44 degrees Celsius and the much lower 32 degrees Celsius. And then both of these temperatures, the bees that were incubated at those temperatures, they kind of displayed a similar survivalship curve here, where there wasn't as significant of a rapid die-off. On to the second experiment involving the measuring of the heat shock protein 70. It was found that the levels of heat shock protein 70 being expressed in the brains and fat bodies were significantly higher following incubation at 46 degrees. These figures are all scaled to relative to the levels of heat shock protein measured in those bees that were incubated at 32 degrees Celsius, so like the room temperature, the average temperature throughout the whole experiment. This further implicates that the high temperature of the bee ball has a negative impact on the life expectancy of individual participants. As for flight muscles, the differences between expression in the flight muscles were not found to be significant, and I will try to offer some reasons as to why later in the discussion. The results for the third experiment are pretty interesting. This one measured bee behavior following a subsequent hornet attack. Both of these pie charts represent bee behavior during the subsequent attack, with the top one here showing bee ball naive participants, of which around a third, or yeah, 1/3 of them joined in on the hot defensive bee ball. And bee ball experienced bees, so ones that participated in the first initial hot defensive bee ball. Around half of these individuals participated in the subsequent defensive bee ball as well. In addition to this, the ones participating for a second time were more likely to fight their way to the center of the bee ball mass, to the core where it is hottest. Likewise, there was also a preference for a central position by bees that were incubated at high temperatures of 46 degrees, but not 44 degrees or 32 degrees. Quick little summary here of what was just discussed. It was found that the hot defensive bee ball, as well as exposure to high levels of heat during incubation, results in an earlier death to participating individuals. High expressions of heat shock protein 70 are observed in the fat bodies and brains of honeybees that are exposed to the same high temperature reached within the bee ball core following their 30-minute incubation. And lastly, honeybees that participate in a hot defensive bee ball were more likely to participate in a subsequent bee ball than bees that had not participated in an initial one and had never been exposed to high temperatures. The authors conclude that Asian honeybees cannot defend their hive from giant hornet attacks without experiencing deleterious consequences on an individual level. Also, an altruistic behavioral change was observed by which bees that were bee ball experienced, and thus going to die sooner, would mitigate the deleterious effects to their sisters by participating in subsequent attacks, therefore mitigating the net loss of otherwise healthy bees. This is an important trade-off as more bees would be available for later defense if needed, as well as for resource gathering. With more bees available to serve these functions, the likelihood of the colony to persist through the winter and for the queen to have sufficient resources to reproduce in the following year will be higher. A good thing to note here is that the worker bees themselves do not have, they don't produce baby bees, I guess. But instead it is the queen that is responsible for this. Thus, this behavior is an example of inclusive fitness, as the benefit to the colony outweighs the cost of expending a couple of sterile workers here and there to maintain the defense of the colony. In my opinion, the study was well representative of a natural scenario in which observation would otherwise be difficult. Obviously, all models have their flaws. No model is perfect, and I just want to go back and discuss some things in the study. So for experiment number 1, part 2, that involved the incubation of bees at the 3 different temperatures and then analysis of their survivalship over time thereafter. I just wanted to point out that a large proportion of the bees died off in the first day following the release back into the hive. And I just want to say this is probably due to stress induced by the experimenters and their handling of the bees to incubate them and interact with them, I guess. And then in experiment number 2, the expression of heat shock protein 70 in the flight muscles was not significantantly different as it was in the brain and the fat bodies. And this is likely just due to the fact that the flight muscles are required for producing that heat needed for a second defense. So instead of the bees just pretty much becoming zombies, all their cells dying and release of the heat shock protein, their flight muscles are reserved for that ability. And yeah, that's my presentation. Thank you for watching. Hope you have a good night. Here's the article if you guys want to go check it out. I do recommend going to check it out. It's very interesting. And if you have any questions, let me know.